Thank you to the chairpersons and thank you to the BEADS committee for the opportunity to present an ongoing study which is truly translational and hypothesis-driven, and the aim is to position microRNA at the center of precision endocrine and surgical oncology. My name is Joshua Agilinko, and I work as a clinical research fellow at the Hammersmith Hospital in the Department of Endocrine Surgery and at the Center of Translation and Experimental Medicine. So we have no conflicts of interest to declare. So surgical management of endocrine cancers is challenged by diagnostic and prognostic limitations, and that is above the clinical as well as the molecular level. And what that leads to is actually lack of robust and signature biomarkers that can help our patients. What we can do is actually use an avenue or a window of opportunity to integrate an overarching in-silico validation, meta-analysis, and experimental data to then identify key microRNAs and molecularly characterize them to treat patients. So in brief, microRNAs are processed in the cellular nucleus. They're then transported out into what is called an RIC. And what that does is then allow cellular degradation to take place, and then that affects target organs such as the endocrine organs. And we know from our own in-silico work, which has been done in the lab, that two main oncogenetic drivers, STAT3, as well as DKK1, which is associated with high-risk adrenocortical cancers, are regulated by microRNAs. Further to that, a recent study looking at 30 cytologically indeterminate thyroid nodules was able to identify microRNA 146B as well as microRNA 375 as key microRNAs that adequately and 100% predicted papillary thyroid cancers and medullary thyroid cancer. And the former, which is miR-146B, was actually able to increase sensitivity of the papillary thyroid tissue to radioactive iodine, and that improves surgical outcomes as well. And even further to that, there is a synergistic and complementary effect of using well-established biomarkers for thyroid cancer, such as BRAF, TERT, etc., in combination with circulating microRNAs, because what that does is at the molecular and clinical level, it improves performance for patients. So our aims are threefold based on such solid proof of concepts. Can we leverage systematic evidence from literature, incorporate machine learning and bioinformatics, and experimentally validate some of the key or putative microRNA genes in the laboratory? So our search strategy is quite simple, straightforward, and it's underpinned by methodological rigor and transparency. And what we ended up with was 38 studies with 4,162 malignant tumors. And this is a 6-stepwise method that we then used to then undertake our machine learning. First of all, we collect our data. We then extract key diagnostic and prognostic algorithms. We then use what is called an unsupervised machine learning and k-means clustering. And what that does is it allows us to then validate some of the key microRNAs into distinct biological clusters. And then we're then able to interpret them at the biological level and also undertake pathway enrichment analysis. I'll share some of the key results now. So when we look at microRNA 221 in particular, and we look at the pooled diagnostic ratio, we're able to see that at a diagnostic odds ratio of 20.2, it has a strong discriminatory power, and that actually outperforms ultrasound scan and is comparable to other targets such as BRAF. And when you proceed to pooled sensitivity, which was almost 90%, again, that outperforms ultrasound scan as well, and cytology for papillary thyroid cancer particularly. And the same thing also applies to positive likelihood ratio as well as negative likelihood ratio. Again, there is an outperformance of ultrasound scan with using a microRNA 221 as a diagnostic marker. When we move towards our prognostic indicators, first of all, when you look at pooled expression of microRNA 221, a dysregulation of that actually doubles your risk of mortality. And in papillary thyroid cancer alone, that is consistent with adverse prognostic outcomes in an overexpressed miR-221. And when you look at overall survival, as well as recurrence. Again, overexpression of miR-221 correlates quite well with worse survival outcomes and increase in recurrence patterns in our patients. And using a machine learning approach and putting all these microRNAs in 6 distinct clusters, and then validating them with targets from 3 large microRNA databases, you can then see from cluster 2 that study count was 23, and miR-221 is in that cluster. And when you then move forward to biologically then interpret them, you can clearly see that not only is miR-221 important diagnostically and prognostically, it's also a pan-endocrine marker, and is able to predict diagnosis and prognosis for thyroid, pancreatic, neuroendocrine tumors, adrenal, as well as parathyroid malignant tumors. And interestingly, when you then do a posterior Bayesian hierarchical analysis, you're able to see that miR-221 has a hazard ratio of 10, and that means it's a dominant and consistent biomarker across all the endocrine tumor. And that also correlates with key oncogenic pathways such as apoptosis, proliferation, and differentiation pathways that we see in cancers, and especially endocrine cancers. And we've then moved this on to the lab as well, and we've looked at incorporating a novel treatment called all-trans-retinoic acid, which is essentially a retinoic acid analog that is able to treat or able to give good outcomes for adrenocortical cancer. And when you do this, you can actually see that when you do target miR-221 and DKK1, as I said, which is an oncogenic driver in ACC, as you increase your miR-221 expression, you actually see a reduction in DKK1 expression. And it will be doing some more work on this in the future. So to conclude, miR-221 I've shown is clearly a pan-endocrine biomarker and is promising with a huge translation potential. And when you incorporate meta-analysis, literature evidence, as well as mechanistic insights from what we have from our experimental data, we hope to look into newer clinical avenues to help our patients. And this also serves as a clear proof of concept, scientifically at least, to provide more data and to provide more work and evidence for going into both in vitro and tissue work. And in combination with other miR targets such as 146B and 375, which we've shown is actually, it works quite well in papillary thyroid cancer. Hopefully we'll be able to do more combinatorial and synergistic studies to see the effect of miR-221 with other existing genomics. Thank you for listening.
Questions? Alison.
So I may have missed this, all right, and this may be a very stupid question. So you've identified something that comes out of all endocrine cells, and you've obviously targeted cancer. It looks like, my understanding is you've targeted cancer within your meta-analysis. Is that correct?
So tissue, 4,000.
So what about the benign endocrine cells? How do you know that this isn't just from an endocrine cell?
Yes, so actually we looked at both malignant and benign tissue. So there were about 4,162 malignant tissues and over 3,000 benign tissues. So these microRNAs are actually differentially expressed in malignant tissue alone.
Sorry, I didn't get it from this. I apologize.
And can I ask, to identify it, is it a simple test that you can do? Is it commercially available? Or is all that on, at the moment, on a research level?
So no, these have been proven. This is all either circulating microRNAs or the tissue itself where you do immunohistochemistry. So this has been shown in vivo and in actual patient tissues as well.
Thank you. That was an awful lot of work. Well done. You should be congratulated.